I am Jasmine Navarez, Digital Content Coordinator at Simulations Plus. I'll be hosting today's webinar behind the scenes, making sure we don't have any technical difficulties. In this webinar, Executive Director of PBPK R&D Haiying Zhou and Scientific Product Specialist Fiona Plait will cover ACATPlus, how ACATPlus can be used mechanistically understand local drug disposition in the gastrointestinal tract. Business Development Director Tom Sanderson will serve as our moderator today. A few housekeeping items before we begin. We take your privacy rights seriously. By registering for and attending this event or participating in the Q&A session, you are allowing us to contact you for follow-up. You may ask questions via the questions panel on your dashboard at any time. If you need assistance, please use the hand raise icon. We'll address all questions in the Q&A after the presentation. And now I'll hand it over to Tom to introduce our speakers.
Thank you, Jasmine. So starting with Haiying. Haiying has a PhD in biomedical engineering from Case Western Reserve University. As Jasmine's mentioned, she's one of our executive directors, in this case for PBPK R&D, where she leads innovation in drug modeling and simulation. Haiying joined Simulations Plus in 2009 and has contributed to development of GastroPlus for PK PD modeling for drug candidates. And she's passionate about applying quantitative systemic approaches to improve drug development and regulatory science. Haiying led the development for ACATPlus through collaboration with multiple pharmaceutical companies, advancing the predictive modeling of drug disposition in local gut tissues. Fiona has been at Simulations Plus for more than 2 years and is one of our scientific product specialists. Prior to joining Simulations Plus, Fiona undertook her PharmD and master's degree in pharmacokinetics at the School of Pharmacy at Paris Descartes University in France. And then she followed up her studies with more than 5 years' experience in PBPK modeling, having helped develop PBPK models in various therapeutic areas for the pharmaceutical industry, the FDA, and other research programs. And with that, I'd like to hand over to Haiying to start the presentation. Thank you, everyone.
Yeah. Thank you, Tom, for the kind introduction. Good morning, good afternoon, everyone. This is Haiying Zhou, and it's really a pleasure to be here with you today. I'm excited to introduce one of the key new features in GPX 10.2. It's ACATPlus module, which brings enhanced capability in our PBPK modeling workflow, allow us to able to simulate some of the local acting compound. Let's first take a quick look at today's agenda. We will start with a brief background and followed by the introduction to ACATPlus, a new module within GPX. We will explore some of the key enhancements, include addition of extra colon segment and more detailed gut tissue model, and also the capability for microbiome-mediated conversion in the gut lumen. Then my colleague Fiona will walk you through a program demonstration. It will use a case study with sulfasalazine and mesalamine. In this case example, it will highlight how microbiome metabolism is implemented and how could we simulate local drug disposition for compounds targeting the colon area. At the end, we will wrap up with a Q&A and open discussion. So for this new function, ACATPlus, this was developed through the collaboration with a few leading pharmaceutical companies. We try to address some of the really need in the industry area to model the local drug disposition. The primary goal of this, through all this collaboration, is to enhance our original ACAT model, by allow a more accurate simulation of the drug distribution in the gut tissue, so beyond the enterocyte. With the original ACAT model, we have the enterocyte layer, but so there are some requests from the industry area. How about as other like the gut tissue, the different locations? Importantly, ACATPlus has also been used in one of the ongoing FDA-funded projects. As we know, for certain local acting compound, plasma concentration cannot really be adequately to reflect the drug level at the target site. So the aim for this FDA-funded project, we try to use ACATPlus module and also try to develop the physiological based biopharmaceutical model PBBM framework to support bioequivalence assessment for this local acting compound in the GI location for both the healthy subject and also the IBD patient. First, let's just take a look at our original ACAT model. So just a review, like it incorporates 9 GI segments and ends at the ascending column. This model really performs very well for simulating like the drug absorption focused on systemic exposure, especially like since most of the absorption may occur in the small intestinal. Even some like a modified administration formulation, it will deliver to ascending column, but it may have good permeability, so the absorption period might not be that long, still able to finish the absorption within like the original, the ACAT, this platform. And also because its primary purpose is to predict absorption, so the model includes only just one single layer of enterocyte cells. So, however, this structure makes it less suitable for simulating like drugs targeting the colon. Normally, to targeting the colon, they either have very low permeability compounds or the formulation was designed to release only in the colon. So for this compound, absorption occurs primarily in the colon and also tends to have a long like absorption duration. Also, this model needs to provide like the drug prediction at the gut tissue level to better predict like the drug concentration at the targeted site. So for these reasons, and we need to make some enhancement to our original ACAT model, and we call it ACATPlus. So let's first look at some of this comparison between our ACATPlus module and ACATPlus. So one of the major improvement is the incorporation of additional like the colon segment. In this slides, we show the examples for human. And we do have like also ACATPlus module like for rat, for mice, and for dog. For human, the total colon, we have more segments. It's not just the ascending colon, it also has transverse colon, descending colon, sigmoid colon, and the rectum. And this is a screenshot like to show the updated ACATPlus human model, and also each this extra segment as the other GI segment, it will have its own physiological parameters. From these slides, we can see the different segment will have its own like the pH, transient time, length, and radius. And also because the whole like the colon transient time is significantly longer. And if we consider the total like the whole colon transient time for human, it will be more than 30 hours, close to 40 hours. And to capture this slow transient, And also it's caused like the regional absorption dynamics to make this prediction more accurately. ACATPlus module also allow user to define the multiple subcompartment for each like a colon segment. Oh, sorry. Like you are not able to set the number subcompartment for small intestinal because the transit time is shorter. But for large intestinal, ascending colon, transverse descending colon until rectum, you do able to change this to like a non like larger than one this number. If you do say, okay, I want to really simulate the distribution at the different locations along the ascending column, you're able to set this to multiple subcompartments. Now let's take a closer look at each GI segment in ACATPlus module. As shown in these pictures, we took it from literature. And each segment include like multiple sublayers, and it will have like mucosa, submucosa, and also the muscle layers. And from another one, like from the ultrasound, you could also see the relationship and of the gut wall. Each wall are different in like thickness and the tissue composition. Like the muscle layer will have a different composition with the mucosa and the submucosa layer. And they also have different blood flow. So the capillary are mainly distributed in the mucosa layers. And the submucosa, we have like the larger artery and the vein goes through these locations. And the muscle also have less blood flow compared with mucosa layers. And this is reasonable because the mucosa layer, this is response to take the drug absorbed through the enterocyte layer and to the blood circulation. By incorporating like this level of anatomical details, ACATPlus module enable like a more physiological accurate simulation of drug distribution within the gut wall. So this is really a critical factor for modeling the GI local acting drugs. Another important factor to consider in modeling local drug disposition is the mucus layer. So the GI epithelium, the layers, is really protected by a continuous mucus layer that prevent the microbiome, like the bacterial access to the intestinal mucosa. The thickness are different from the different locations, the GI locations. Like the measurement in rat shows, like in rat stomach, it's between like 100 to 300 micron based on the different locations of the stomach. And for the intestinal, the small intestinal might be close like 100 micron, but in large intestinal, it's much thicker. It could be around 900 micron. And also the structure are different. In the stomach and the colon, it normally will have like 2 layers of mucus. One layer is loosely like attached layers, it's at the outside. And another is firmly attached, like inner layers. It's firmly attached to the mucosa layer. It's very difficult to remove. For the loosely attached, maybe you rinse with some water, you're able to remove the loosely attached mucus layer. But the firmly attached is really like especially the attached part to the mucosa part is difficult to remove. And if you really try to completely remove it, you might cause damage like to the mucosa cell. For the small intestinal, and the firmly attached layer is very thin. And sometimes you can think it as a single mucus layer, and most is loosely attached. So normally, in normal conditions, typically it's not attached to the mucus layer, and it's not continuous. And another important one is the the inner mucus layer is like continuous regenerate. And the measurement from the mirroring the distal colon tissue, and when they try to remove the mucus layer and try to measure how it's regenerated, like for this in the colon, it could be the 1 to 2 hours. It's a turnover rate. It's regenerate the mucus layer. Yeah. So this is like to consider all these above factors, and the ACATPlus module, we need to introduce a more detailed like the gut tissue structure to be consistent like with this picture shows like the different layers, and we also add the mucus layer, and we do enhance our original ACAT model to include 4 extra layers additional to the anterior side. We have the mucus, mucosa, submucosa, and the muscle layers. And each layers like based on the properties, they will have different thickness and different blood flow. And for the mucus layer, the drug need to diffuse through the mucus layer to reach the anterior side and get absorbed. And these diagrams like illustrated like the different layers we incorporated to the ACATPlus module. And also with this enhanced model, it's allow us to simulate the drug concentration in each individual like layers. So normally these are difficult to measure experimentally. Like we could provide like the concentration in mucosa, submucosa, or even lumped like mucosa, submucosa. As I know, during our model development, the sponsors do try to separate like the layers, but the mucosa and the submucosa layers are together, but they separate the muscle layer separately. Or they could provide the simulation of the entire gut tissue compartment, which will represent a typical like a tissue biopsy. So if you take a biopsy, that's really be a lump of the like the different layers, and there will be also maybe some like a mucosa layer, and also the underlying tissues. The next slide just show a screenshot, shows the different physiological parameters of each tissue layer at the different locations, such as like the mucus thickness, like the mucosa thickness and the mucosa blood flow, all these physiological parameters. So you could also set this by yourself, modify these parameters if there are some disease conditions. In addition to the structure, like the layer, different extra structure layers of the gut wall tissues, the mucosa layer also play a critical role in the drug disposition, how the drug get absorbed. This imaging shows the variation in the mucus thickness across the right GI tract. You can see in the small intestinal, like it's thinner, much thinner, and also the firmly attached mucus layer, it's much thinner. But if we look at the large intestinal, especially in the colon, it's like significantly thicker compared like to the small intestinal. So, and also like to accurately model the drug transit through this barrier. Like this is on top, this mucus layer is on top of the enterocyte. This is facing the lumen content. To better simulate the drug transit through this mucus layer, this barrier, there are several factors we need to consider. This include the compound diffusion, diffusivity through the mucus layer. And is there any binding interactions within the mucus layer? And also the regeneration, on the surface, on top of the mucosa, there are cells will continually regenerate the mucus. This regeneration will push the mucus move up, and at the top it will shed off. So to better describe this mucus layer, we need to consider all these key factors. And this is how we're handling this. In the mucus layer of the model, to accurately simulate the drug movement through the mucus barrier. And because it's much thicker compared to a single layer of cell, we could use well mixed, but this is so thicker. And in able to simulate the delay, we incorporate the multiple sublayer structure for the mucus layer. So currently, it's divided the mucus layer in the program, divided to 20 sublayers to capture the distribution delay from the lumen to the enterocyte surface. This is mainly caused by the compound diffusivity in the mucus layer. In the mucus layer, the diffusivity will be smaller compared the diffusivity in the water. And also it will have some bindings. All this you could do obtain these values through experiment measurement. Also, as I mentioned, it's also considered several key physiological mechanisms and the diffusion from the top to the bottom. So this will be from the top is the lumen facing the layer, and the bottom layer is the enterocyte, is the epithelium facing layers. And at each different locations, it will have the location specific rate for the mucus regeneration. At the small intestinal, large intestinal, and also stomach, they will have their own the mucus regeneration rate. And because of this regeneration, the mucus renew will push the mucus from the bottom to the top, there will be upward cause the compound upward transit. When it's reached to the top layer, and it will be shed off, and from the top layer to the lumen. So all these mechanisms are very important to model the local drug disposition, especially for this compound targeting at the colon. The next very important aspect of the local drug disposition is the microbiome-mediated metabolism. So in ACATPlus module, we incorporate the nonlinear, the kinetic model to simulate the drug conversion by the gut microbiome. And you could set it through the enzyme table, and we use microbiome, and the location will be the lumen. It will only convert the dissolved compound, and with certain Vmax and Km. Even though most of the conditions, you may use the fecal data, use the feces data. What you measure is maybe half time, but you could use a higher Km to convert it to a non-saturable, a kinetic model, and convert a Vmax for these reactions. And the model to provide, the program to provide the default microbiome expression along the GI tract. And you can find it's mainly distributed in the large intestinal. This is just try to be consistent with the measurement data. The microbiome distribution primarily located in the lumen of the large intestinal. So the small intestinal, the microbiome distribution is orders like smaller. And this capability is very important, very useful to simulate like a drug, like sulfasalazine. So this will be a case example we will show today, which undergoes like extensive microbiome conversion in the colon. These are like, I just introduced the previous couple of slides. I just introduced some of the key mechanism for the enhancement we incorporate to the ACATPlus module. This is a slide I want to summarize the key mechanistic enhancement we introduced in ACATPlus module compared with the original ACAT model. So first, the gut tissue structure has been expanded to include 4 additional layers, and this we incorporate mucus, mucosa, submucosa, and also the muscularis layers. This will be allow for more detailed simulation of the drug distribution within the gut wall tissue. And second, we also include the 5 colon segments and ascending colon transverse, descending colon sigmoid, and rectum. This is for human, to provide like more detailed anatomical like resolution, so able to simulate the longer absorption duration in colon, and to better capture like the slow transient and also the regional absorption dynamics within the colon segment. And we also provide the user flexibility to define multiple subcompartments for each colon segment. And at the last, the microbiome mediated conversion really enable like the precise prediction of the microbiome metabolism across the different intestinal region, and for those compounds will undergo like the microbiome conversions. Yeah, that would be like some of the mechanism I want to the brief introduced today. And the next, my colleague Fiona will give a demonstration of the case study. Yeah, Jasmine, you can show the case study.
Sulfasalazine is a low permeability compound, which is metabolized by the gut microbiome in the colon lumen into mesalamine, a drug that also exerts its effects in the colon and rectum. The sulfapyridine is the second metabolite formed in the colon, which is non-active. Let's now switch to the GPX to see first how to implement the microbiome into the sulfasalazine model. Currently, we are in the sulfasalazine and mesalamine example project that we will use in this tutorial. This example project has already been fully set up, except for the microbiome enzyme that we will add. Let's move to the compounds view. Here, we will select the sulfasalazine compound if not already selected. You can review the inputs in each panel by clicking on them. And you will see that it has been already set up with everything. Now we will open the enzyme kinetics panel. We will now add the microbiome by clicking on Add. So here we will leave the species as human, and we will also leave the wild phenotype. Under the enzyme column, we will select the microbiome. For the location, we will select lumen dissolved. We will also change the Vmax and Km values. We will enter a Km of 20 milligram per liter and a Vmax of 0.005 milligram per second. Now you will click on Save at the top right of your project and then on OK. Don't forget to save your project frequently. Now let's move to the physiology's view. Make sure that sulfasalazine is the selected compound. You will see one physiology available, which is a physiology for a human of 30 years old and 70 kilograms. Now we will open this gastrointestinal panel. And if you scroll down a little, you will see that the ACATPlus is enabled. It has been toggled on. And if you scroll down again a little bit, you will indeed see that you have the extra colon segment present here. So you will find the transverse, descending, and sigmoid colon, as well as the rectum. If you again scroll down, you will see after the ACAT plus physiological parameters table, the enzyme distribution, in our case, the microbiome lumen dissolved expression. As you can see, there's no expression in the upper part of the gastrointestinal tract, and it's in the colon part where you will see that we have a microbiome expression. Now, let's move to the pharmacokinetics view. Make sure that the sulfasalazine compound is the one selected, and then you can click on the compartmental panel where you will see that a two-compartmental model has been entered. This model has been created and validated based on preclinical IV data. Now let's click on the simulations view. We will select the HVPO sulfa 1 gram suspension, if not already selected, and click on Run Simulation here. Once the simulation is done, the software will automatically switch to the analysis view. And you can observe that the simulated CP time matches the observed data pretty well for a single dose of 1,000 milligrams of sulfasalazine administered to healthy subjects as a suspension. Now click again on the simulations view. We will run another simulation, which is the HVPO sulfa 2 gram and coat tab, and then click on Run Simulation. Again, once the simulation is done, you will see the analysis view with the CP time. And you can see here that the simulated CP time is reasonable compared to the observed data for a single dose of 2,000 milligrams of sulfasalazine administered to healthy subjects as an enteric-coated tablet. We will now move back to the compounds view and select another compound, which is the one called sulfasalazine with MET. In this case, it's the exact same setup of the sulfasalazine compound. The only difference, as you can see here under the enzyme kinetics panel, is that the metabolite is now entered. So we entered the mesalamine instead of none, as it was previously the case. So this represents the sulfasalazine being metabolized by the microbiome into the mesalamine in the gut lumen. We will now simulate both sulfasalazine and mesalazine CP time profiles. Click on the simulations view. And then we will click on the simulation, which is called HVPO MESA 2 gram enteric coated tab, and click on Run Simulation. Because the simulation contains the ACATPlus model, the time for the simulation to complete can take longer than usual, depending on your computer's power. Again, once in the analysis view, you will see the simulated CP time curve for mesalamine and sulfasalazine for a single dose of 2,000 milligrams of sulfasalazine administered to healthy subjects as an enteric coated tablet. Now, to be able to distinguish both compounds, let's expand the additional features by clicking on the plus here. You will select the compound and then click on Color By. Now, let's put the plot on the semi-log scale to have a better view of the mesalamine simulated and observed concentrations that appear now in green. We will click on the minus for the additional features and expand the plot settings by clicking on this plus, and then the plot options by clicking again on the plus. If needed, you can expand the plot settings and plot options by dragging to the left here the plot. Now you will move to the dependent axis, that is the concentration, and change this to logarithmic. You can close it by clicking on the minus here, and you will see the mesalamine observed data and simulated concentration. Click on the observed data. You can see under the summary of data profiles that we have 3 exposure data type. And as you can see, those 3 correspond to the observed concentrations for the 3 simulations that we just ran. So you can see 2 exposure data corresponding to sulfasalazine concentration for a 1 gram suspension administration and for a 2 gram enteric coated tablet administration. The last one corresponds to mesalamine observed concentration after a 2 gram enteric coated tablet administration of sulfasalazine. We also have 1 in vitro dissolution release, as you can see here. And so let's select it by selecting the in vitro dissolution release data type in the drop-down menu. If you scroll down, you will see the dissolution data fitted by the Weibull function for ACAT. ACAT is the brand name for one of the mesalamine delayed release tablets. And in the study, patients received 1,200 milligrams of ACAT per day during a week to reach the steady state. On day 8, biopsies were done on those patients. So we will now enter the rectum concentration that was measured with those biopsies. So let's move back up and select again the exposure data as data type. Click on Add next to the group name, and you will enter the name ACAT gut tissue concentration. Then click OK. You will then click on the Add button next to measurement type, and you will call it rectum concentration. You can then click on OK. Once you are in the rectum concentration, you will change the concentration from nanogram per ml to microgram per ml. Now you will enter a time point of 176.5 hours, which corresponds to the data point when the biopsy was performed, and then enter a concentration of 108.5 microgram per ml. You can now see on the plot the measured observed data that you just implemented. Now, if you scroll down, you will see a table where you will select gastrointestinal tract for the module. Under section, you will select the rectum. Under compartments, you will select the gut tissue. And as you can see, the state is already filled with the concentration present. Now, we will save it and click on OK once it's saved. Now, let's move to the simulations view. We will select the simulation called Patients 400 milligram times 3, 1 week CR tab as a cold DVOS. Once it's selected, you will click on the Run Simulation button. So once the simulation is complete, the analysis view will appear and you will see the simulated CP time of mesalamine at steady state. You will now change the mode that is currently as the key view mode to the deep view. You will then expand the mesalamine by clicking on the gray arrow, then gastrointestinal tract, concentration, gut tissue, and here you can see that all of the ACATPlus segments are available. In our case, we will select segments from the cecum to the rectum. You can see that now concentration present is available for selection. So you will click on it and you will see the mesalamine gut tissue concentration for all the ACATPlus segments that we just selected on the plot. Now we will add the measured rectum concentration that we just added into the observed data. To do so, we will expand the exposure data here by clicking on the arrow. Then click on the ACAT gut tissue concentration and click on the concentration series. You will see the rectum concentration appear. To select it, you will hold down Control and select the rectum concentration. Now both measured and simulated concentrations are selected. Let's now differentiate the rectum concentrations from the other segments by coloring it. To do so, expand the plot settings by clicking on the plus. Now you will select the measured rectum concentration and also the simulated one. So here you can hover over to see the entire name. And here you can see rectum. So you will hold down Control and select this. Now that the rectum concentrations are selected, you can click on the plus on the plot options. Again, to be able to see the entire window, you can drag to the left the plot. Here you will scroll down and under plot options, you will select the square next to options to select another color than black, for example, red. Then you will click on OK. You will then select this other square next to line options and the points and select red as well. Click OK. You can now click on the minus of the plot settings to see the plot, and now you can see in red the measured concentration in the rectum and the simulated one. As you can see, the measured rectum concentration is in the same rank as the simulated concentration. You can also unselect the other segments by unchecking them to be able to focus only on the rectum concentration. So to do so, you will uncheck the other segments. Here, you will now only see on the plot the rectum observed and simulated concentrations. That brings us to the end of the ACATPlus tutorial. Thank you for watching. If you would like more information, please visit our website at www.simulations-plus.com, or you can email us. Thank you.
And hand it over to Tom for the Q&A.
Thank you, Jasmine, and thank you, Haiying and Fiona, for a great presentation. So we have a few questions that have come in, and please do keep typing them in. And if we can't get to you, we'll obviously share with you afterwards. The first question I have is, can subcompartments in the colon be defined as representing certain colonic diseases?
Yeah, I will take up this. Yeah, that's really a good question. Like currently, like for the subcompartment we use, it's mainly try to simulate the transient dynamics, the slow transient. So they will share the same physiology, like in the same like ascending colon, descending colon. Even though they divided to like the 10 subcompartment, but their pH, like the biosalt, all this will be same, and like the radius and the length definitely will be divided by 10. We assume they're evenly divided. We haven't used like to represent the disease condition specifically at a certain subcompartment. Currently, through the collaboration with FDA, we do develop like IBD models, like for Crohn's disease, for all the different disease conditions. For all that, we assume any physiology change is happen to across the whole segment. We haven't used the subcompartment for that. Subcompartment will be mainly used to simulate the transient and absorption dynamics. Yes.
Great, thank you. Another is how to calculate the Vmax and Km for relevant microbiomes.
Yeah, that's really that's a good question. Yeah, it really depends on like your measurement. As I know, currently most of the conditions is really like use fecal data, like they dilute the feces, and then they measure the stability of the compound in the feces. And what we are using currently, we use a conversion is we assume the microbiome in the feces sample you have will be similar as in the large intestinal like lumen. If we don't know like the microbiome like numbers, the difference, and we really couldn't do the conversion. Currently, assumption is assume like in the fecal sample you use, and then the lumen content, you just assume, okay, 1 gram of fecal material in the in vitro data you use, and also 1 gram of lumen content, we assume their microbiome content, the density are same. The only difference is because of the dilution. And in this way, we could convert this stability data half time like to clearance. And then we assume like non-linear, like non-saturable like clearance. So we just give it a large Km and then converted this non-linear clearance to Vmax. Yeah, based on this non-saturable Km. Yes. This is, I think this is also come to the next questions. Was the Vmax and Km calculated using an in vitro experiment or the in vivo studies? It could be from the in vitro experiment, like the in vitro one, if it's fecal data, we could convert it. But also, if you do know, like your both the parent compound and also the metabolite, like converted by the microbiome, and if you have any of the studies available, clinical study available, you could also fit the data, optimize like a Vmax and a Km. Yeah, it can come from both. Yeah.
Thank you, Haiying. And then another question is, can the updated version of the ACATPlus predict the effect of a prokinetic drug on gastrointestinal transit time in a drug-drug interaction study?
That's really like if you're like the it won't affect because your model is mainly if you like your drug-drug interaction, like it depends on where it's happened. It's mainly will like give good prediction for the compound has been mainly delivered like to the colon. It won't affect the DDI because it's still like what happened in the liver, it won't affect. And what happened like in the colon, compared with the original model, there might be some difference because now your drug need to go through like this mucus layer and able to reach like the colon tissue. But this is normally if your previous model, you even use original ACAT model, you might need to think some way like to how to better simulate like the colon absorption. As I know, some of the users they need to simulate like the colon absorption, prolonged absorption. They need to like modify the colon transit time. Actually try to mimic currently what we use is ACATPlus like module. I would say as long as your model is validated with the experiment data, it won't affect the DDI. But you cannot just directly use your originally developed like ACAT model and then directly use it with ACATPlus. If it's mainly absorbed from the small intestinal, there won't be significant difference because the mucus layer is pretty thin in the small intestinal. And we won't suggest you like, okay, you need to use ACATPlus module. You need to based on your applications and also your compound. You need for some of this compound, if you still like continually mainly absorbed through small intestinal, and you may still use the original ACAT model. It won't affect like any of the DDI or any other applications. The application is will be mainly like if your drug targeted at the colon or it has long absorption period from the colon, and you will choose to use ACATPlus module instead of original ACAT, because even you try to use original ACAT, most of the conditions you may need to modify the like ACAT module, the original ACAT. You may use a longer like ascending colon transit time to make sure it has enough time to get absorbed from the large intestinal. Yeah, generally like it depends on your compound. If your compound like build with ACAT plus, and you will use just the same model for the DDI. Yes.
Great, thank you. Well, I think in the interest of time, just to make sure we don't go into other people's meetings or so, I'd like to hand over to Jasmine. Thank you, Haiying. Thank you, Fiona, for a great presentation.
Thank you all. For any questions we were unable to answer live today, someone from our BD team will reach out to you with answers following the webinar. We invite you to visit our website to learn more about GPX.2 and our other software platforms. You can also follow us on social media. I put those links in the chat. This webinar has been recorded for playback and will be available on our website and YouTube channel. This concludes our webinar for today. Thank you, everyone.